Hello, this is the part 2 of chapter 19 viruses, and now we're going to talk about retroviruses. And these are a few things that you need to know about retroviruses. This virus is very interesting because what it does is actually have RNA information, and once it's inside the host, it actually asks the host to basically translate RNA information into DNA, and then basically it insert its own viral DNA into the host DNA in the chromosome. And from that, the host will actually go through the normal process and create the viruses part, different viruses parts using that newly inserted DNA. So it's very interesting because it almost tricks the host to make more things for itself. So one of the examples is HIV, which we'll be talking about now. So HIV is a retrovirus, like we talked about. It has besides the information, the nucleic acid information, besides that capsule, it also have this membrane. So it can fuse with the host cell, and then from the viral RNA, it will actually be using a specialized protein that it brings with it. It's called reverse transcriptase or transcriptase. It depends on how you want to say it. What it does is actually changing the RNA into a DNA, and by doing that, it actually would create a new DNA strain, double strain, and then that DNA will be inserted into the nucleus of the host cell, and then it will basically allow itself to attach to the chromosome DNA of the host cell. And from there, normal things happen, transcription happen, and then you have the newly synthesized mRNA, and that will either become the coat, the envelope outside, the coat, the capsule, or the mRNA itself. Well, the mRNA would eventually be isolated and then join with the envelope, and then alongside with the capsule, and then be newly released into a new viruses like this. Okay, so this is how the HIV viruses looks like. So these two pictures are actually talking about when HIV entering the cell. So it has the envelope outside, so it basically fuse with this host cell, and what it does, it would release a membrane, like we show you right here. The membrane would basically go into the membrane of the host cell, and then from that you'll be seeing all these information basically literally fuse into the cell itself. So that's what you see. Okay, this is white blood cell, which is the main problem of HIV, because we won't be able to use our immune system because white blood cell won't be able to recognize that one is a foreign object. So this one is new HIV synthesized from the host cell leaving the host cell. So you have the budding, and then as you can see, the material is gathering right here, and eventually budding completely, and then it fuse out completely by itself to form a new HIV virus. Okay, so a few things I forgot to mention. Proviruses is actually the DNA that's inserted into the chromosome. So basically, this is going to become the viruses. That's why we call it proviruses. And when you hear about the terminology of HIV positive or negative, HIV positive means it's confirmed that you do have a person has the HIV DNA inserted into their host cell. So that's called HIV positive. And then furthermore, you also hear the word AIDS. Well, the word AIDS actually means the viruses has actively start to reproduce itself. So you start having a new HIV viruses, new retroviruses leaving the white blood cell. That's what those two terminology means. And again, I'll show you the video. I'll give you the link. And some of the other viruses on the left hand side, it's herpes viruses, and the two different strain. This one you can see from the mouth, and this one you can see from the genitalia, and then they're basically similar. Oh, sorry. So from here you can see they have very similar symptoms. However, there are two different types of herpes viruses. On the right hand side, we have the smallpox viruses. This has been eradicated, which means we basically completely eliminated from it to revive again. The new smallpox viruses basically are completely not heard of, and because the worldwide organization actually vaccinate every single person that has smallpox, or to kind of like to prevent the possibility of having smallpox coming out again. So, so these are the two other example of human viruses that we encounter. Furthermore, we hear this. This is quite popular nowadays. You have Ebola, and Ebola is actually a viruses that can basically transmit through bodily fluid, and then many symptoms are following. You have fatigue, fever, and it basically have very similar symptoms as a very severe cold plus some kind of food poisoning. So this is very hard to detect, and then because it actually can have the symptoms from day 2 all the way to day 21. So that's why if you go back to a few weeks before and to see or go on internet to do a search, you will see that they have a quarantine period to make sure this virus is not going to activate itself in from the host body. So there is actually not a lot of treatment for Ebola. The followings are listed. So we have some intravenous fluid that we can basically introduce into human, we basically provide a very balanced electrolytes to basically allow the human system, immune system, to catch up and then to fight the viruses. There are some experimental drugs. There are some antiviral drugs, and then some transfusion, plasma transfusion, from the survival, and then with different kind of antibody that's made from this company. And there's actually no vaccination yet. There are some of experimental vaccination, but there's not a completely vaccinated prevention for Ebola. And these are some of the stats that is out there and that you can find as well on internet. This is this number of death. It's not fully updated. So when you see the video at the time, if you would like to find out the more updated number, you can actually go ahead and find that out in the worldwide World Health Organization. And also, it's in comparison, just draw a comparison. The seasonal influenza, common cold, actually has a lot more lethal effect on human life, and we don't talk about it often. So it's actually very similar to this strain as well. So this is just a stat to kind of compare the seasonal influenza viruses versus Ebola viruses. However, I'm not saying Ebola virus is not powerful. It's very lethal. It's very deadly. So there is still very severe consequences to that as well, and as well as influenza that we see. Just basically stay healthy, and if you're sick, just stay home. So this is the map that shows you the outbreak and the concentration of the Ebola outbreak during 2014. So you can see it covers 3 different nations. You have Sierra Leone, much higher concentration in Sierra Leone, and also Libya, not Libya, Liberia, and you have the Guinea. So all these are basically very heavily affected nations around that is affected by the Ebola outbreak. And these are just some statistics of reporting of different things, and then it's just crazy to see how, even though it's really far away from us, but it's actually very relevant to our every single day life, because it's just a viruses that's so small yet it has such powerful effect in human society. So some of the way Like I said, there's actually no vaccination for Ebola, but some of the way to prevent viruses is actually provide a vaccination. So vaccination are weakened viruses, so we basically kind of tone down its ability, potentness, and then also some of the pathogen parts to allow our immune system to basically catch up and then be able to react and respond to the viruses when it actually attacks us. So basically, we're training our immune system to become better at detecting things. Some of the viruses that's quite prevalent is one of them. Well, let's talk about the old one. So 1918, you have the flu pandemic. So once a while, these viruses will mutate into something much more lethal than the other. So some year it will be much more powerful, or some year it would not be as powerful. And so I think you guys should know, if you guys Google it, H1N1 influenza A viruses. This was a huge pandemic situation in 2009, and it caused worldwide alarm. It's actually come from influenza viruses, so it's very similar to our common cold, but it's a such potent mutation of this the blue part. These are the viruses that it causes such severe consequences like death. So that's just from the mutation of a single viruses. And then this is another thing that will actually virials, and let me say it correctly, viroid. Viroids are actually something that causes plants to get sick. So they're very similar to viruses, and but they don't basically affect animal cells. They affect plants. So for this one, it's a viroid that causes coconut plants to basically not grow properly. So as you can see, the affected one falls, all the leaves falls. So it doesn't allow the plants to grow, and it regulates the whole system within the cell to basically become non-functional. And this is another type that's very potent. This is prion. Also, you can call it prion. So what it does, it's actually it's not a viruses. It's a protein that doesn't. Well, if you will, you can almost think of it. It's a protein that doesn't know how to fold itself. So it's a misfolded protein, but it's very potent like a viruses. It will actually cause a normal protein to become abnormal. So this picture kind of helps to illustrate my point. This is a prion or prion. It's a protein that is misfolded, so it doesn't properly work its way. So from this protein right here, you can actually, this protein will go ahead and infect this normal protein, and what it does, it changes shape. It changes normal protein shape into this new prion's shape, and then furthermore, they're gonna affect another protein, and so on and so forth. Eventually, you have a huge aggregate of a prion, so all misfolded protein. So what happened? It destroyed the normal property of a protein. Some of the examples are mad cow disease. This is one of the most interesting disease that actually affects us very closely. And then there are some other disease that we also know. So what it does is it basically changes the protein function, so it doesn't allow its normal function to take place. So what happened is prion, because it does it slowly, it have to affect individual protein. It actually has an incubation period of very long time, at least 10 years before the symptoms can be developed. So mad cow disease, the same way, if a person eats mad cow, so that prion can possibly turn that person into something that has mad cow disease symptom. So there's actually no cure for prion disease, because we have not yet determined a treatment for prion. And right here it's a picture of a person with prion disease. So what happened with this person is, once the symptom is observed, basically this person's brain would actually start shrinking